Good afternoon, my name is John Carlett. I've been working with Dr. Wallace for over 2 years now, and our main focus of our research is basically on cell migration, and we try to find genes that affect cell migration. So cell migration is important because it's needed for the development and maintenance in organisms. The wave-scar pathway is one of the pathways we focus on in cell migration. So the way the wave-scar pathway works is that when a signal comes and attaches to a receptor, it turns on SED10-RAC1, which is a GTPase, it's basically a switch. So this activates GEX2, GEX3, and eventually ARP2-3. And ARP2-3 activates branch-actin nucleation. So that helps with cell movement. And usually in humans, when the wave-scar pathway is affected, it can lead to breast cancer or other neuronal diseases, and sometimes they can even lead to schizophrenia. But back to C. elegans, the way wave-scar works in C. elegans is that it helps with the migration of the epidermis. So when there's a mutation in the wave-scar pathway, it leads to a phenotype called GEX, which means that the intestines are outside of the embryo. And the reason why we use C. elegans is because, one, they're small, so we can house a lot of them. Two, their life cycle is very short, it's 3 days. And 40% of the genes are relatable to humans, so it really makes a good impact. In our research, we use a mutant C. elegans that's called SAX3. So SAX3 is a gene that works in the wave-scar pathway, and when that's mutated, it leads to 45% lethality. So 45% of the embryos that we collect from SAX3 worms are usually dead. And when we introduce RNAi to silence other genes in this pathway, we try to look for enhancers or suppressors. And one of the main RNAis that we used was HUM7, which led to a 23% lethality. And that was significant to us because from 45 to 23% was a huge jump. So this is the domain of HUM7. So it has myosin motor and ROGAP. ROGAP is important because it's a GTPase. It works with the GTPase by turning it off. So when HUM7 is mutated, it can lead to these phenotypes right here. So you see how some of the embryos are deformed or they're missing a whole chunk, which is pretty important to development to the adult stage. And basically from the research, from here on out, like our future directions with the research is to finish the screens, because we did almost like 1,500 screens with the RNAi. We want to finish the rest of the 500, but with the other strains, we finished the 2,000 RNAi screens, which was a big accomplishment for us. So thank you.